Namaste students. I am Chiran Dr. Aziz Mohammed Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapith Women's University, Jaipur. Today I am going to discuss about the B cell activation. So this topic is belonging to immunology, basic immunology, principle of immunology. In the last video, I discussed about the activation of T cells and T cell activation can also help in the activation of the B cell. So here we will discuss about the B cell activation. What are the B cells? B cells are the cells which are originate in the bone marrow, and after their origin, they get mature in the bone marrow also. B cell have some receptors which are known as the B cell receptors. B cell leave the bone marrow and circulate between the blood, the secondary lymphoid tissue and the lymph, so that they can identify a particular kind of antigen. They can identify the antigens. They can engulf the antigens because the B cells are also antigen presenting cells. Like another antigen presenting cells, B cells are also antigen presenting cells. We talk about the B cell activation. B cell activation are of two types. It is T cell independent or it is T cell dependent. So first we will discuss about the T cell independent B cell activation, how it goes. First thing, the B cell activation in the T independent activation is that first the B cell will identify it is a antigen presenting cell, so it will identify some antigenic receptors and will bind to the antigenic receptors on the surface of the bacteria. Different, there may be different antigenic epitopes will be there, and they will bind to those epitopes by the B cell receptors. And after that, the clustering of B cell receptor will trigger the B cell activation. So after that, some they will capture the antigen epitopes, and the toll-like receptor will come. It was the first signal where the clustering of the B cell was conducted, where it was identifying the epitopes of the antigen and binding to epitopes by the B cell receptor. It was the first signal. The second signal is conducted by the toll-like receptors. After binding to the B cell receptor to the antigens and epitopes, the toll-like receptor will also bind to some epitopes of the antigens. Toll-like receptor will identify some particular pathogen-associated recognition motifs, and it will bind to that uh, antigen. So, and this will also activate the B cells. After the signaling, B cell get activated and it will convert into the plasma cells. It will differentiate, it will proliferate, and it will differentiate into the plasma cells. Each plasma cells will then secrete some immunoglobulins like IgM, IgG. Okay, so this is in the B cell activation. This is T cell independent. In the T cell independent B cell activation, there is not any kind of memory cells. So I must share the screen here. So in the first step, B cell receptor will bind to a particular epitope which is present on the surface of the T uh, antigen, which is present on the surface of the antigen. it is the first signal. After that, the toll-like receptor will also bind on the surface of the antigen. Okay. Toll-like receptor will identify some pathogen-associated molecular motifs, and these two signals will activate the B cells. And after activation of the B cell will proliferate and it will differentiate into the plasma cell and secrete some specific antibodies against some the pathogens, against the bacteria or the microbes. So this is the T cell independent activation. Now this is the summary of the T cell independent activation. Now we talk about the T cell dependent activation of the B cell. In the first signal is the B cell and it will capture an antigen. This is the B cell and it is a antigen presenting cell. So it will capture an antigen here. That B cell will present this antigen on the surface by major histocompatibility complex. Okay. It will process the antigen, it will phagocytose the antigen and process, and after processing it will make some digestion on the antigens, and then it will present this antigen on its surface by the major histocompatibility complex. Then it will also have a co-stimulatory uh, co-stimulatory receptor. Some so it is presenting here. The same antigen was also encountered by the T helper cell. Okay, same type of antigen was already encountered by the T helper cell. So that T helper cell will come and it will bind. It will bind to the MHC molecule. So this MHC molecule will bind CD4 CD4 receptor will bind to the MHC molecule, and this antigenic receptor will bind to the antigen. Then T helper cell is bound here. This is the first signal of the T helper cell. After that, a co-stimulatory signal will be there. This co-stimulatory signal is conducted by the CD40 ligand, CD40 receptor. So CD40 receptor and the CD40 ligand of the T helper cell. So this T helper cell has some ligand. It is sending some message to the B cell, and the receptor of the B cell, that is CD40 receptor, will receive this signal and it will get activated. After activation, the B cell will get proliferated in various numbers, and after proliferation the B cell will differentiate into the plasma cells. After differentiation into the plasma cells, the B cell will also differentiate into the memory B cells, so that the memory of a particular antigen uh, can be kept and stored in the immune system, so that when the same antigen will encounter into the immune system, then the B cell will take lesser time to respond. So memory cells are prepared here, and now uh, they will convert into the memory cells and the plasma cells. These plasma cells will now further convert it into the antibodies. They will secrete some antibodies. So first we are completing the complete lecture here. First of all, antigen is recognized by the B cell, and it will process the antigen and present this antigen on the surface of B cell by major histocompatibility complex number 2. This major histocompatibility complex will attract the T helper cells. The T helper cells will come, and T helper cell will bind to the CD4 receptor of the major histocompatibility complex, and the another portion of the T helper cell receptor will bind to the MHC class 2 molecule. This is the signal number 2. After that, a co-stimulatory signal will be there, where the T helper cell ligand will send the signal to the CD40 receptor of the B cell. The CD40 receptor will receive the signal and will get activated. And after that, the interleukin 4 will be released, and these interleukin 4 will come and bind to the B cell. This interleukin 4 will then activate the B cells. The cytokine receptor will activate the B cells, and this activation will lead to the proliferation of the B cells. After proliferation, the B cell will differentiate into the two types of cells, and the plasma cells and the memory cells. The plasma cells will further differentiate and convert into the antibody. They will secrete antibodies, and the memory cell will store the message, the signals from the antigens if they were further encountered in the next time. So B cell is converted, differentiate into the plasma cells, and will secrete the antibodies in a single seconds. So here, I hope the students will able to understand the process. So in this process, the B cell, in this video, the B cell activation was explained, where the B cell activation on the T cell independent and the T cell dependent mechanism was explained. I hope students will better understand this by the diagrammatic methods, and some questions which can be asked in this lecture are: the first one is, what is the mechanism of B cell activation? Explain the mechanism of B cell activation with diagrammatic representation. Explain the T cell independent and the T cell dependent B cell activation. So I hope the students will be better understand. So my lecture is over here. Thank you very much.